Paul, thank you so much for joining me here on Wilder Future. I am so pleased to have you on the show, and yeah, I can't wait to get into it. Could you just do a little introduction about your background and what you're into, and what you're doing right now?
Well, my background is in microbiology, specifically anaerobic microbiology. I became fascinated by this topic when I was in college, and I kind of came to the conclusion I was a biology major, and it kind of hit me that microbes were really the center of everything. They were responsible for really so much of what was going on in the world, and it was all going on unseen to us. And I was always raised that microbes were kind of, they were germs, they were bad for you. And it wasn't until I really got started reading more deeply into microbiology that I began to appreciate the fact that there's way more good bugs than there are bad bugs, and they really underlie all the global biogeochemical cycles. They clean the air we breathe, they purify our water, they recycle our waste. They're involved in so much, really throughout geological history, and it became sort of a fascination with me. And then I became interested also in how we interacted with microbes on a sort of, I guess you could say, environmental level. And I was fascinated by the fact that there were all these microbes out there that could live by not breathing oxygen like we do. And so as I delved deeper into this, looked at the metabolism, and then started looking at environments where these were the most important agents. And one of them was the rumen of ruminant animals. And I became fascinated by the fact that the cow could not live without its microbes, that the microbes were really responsible for digesting all the feed that the animal was consuming. And so I sort of gravitated toward this as a career path, and that's where I ended up. So it's been an interesting journey. I've learned a lot, and the nice thing about working with organisms like cows is you start to interact with all kinds of people who look at cows from different perspectives. You have producers, you have animal nutritionists, you have people who handle the manure. You work with people that are concerned how you feed them the best, and then of course with the economists and everything who have to make dairying profitable for the farmers. So you get a chance to see a lot of different perspectives. And I kind of value that. So that's kind of where I am.
I couldn't agree with you more. And what's so exciting for me is that you've done primary research into this incredibly important area. When you hear fermentation, or probiotics is a great term people throw around.
Right.
And it's sort of like saying life, because it's just such a broad category, and it's just so deep when you get into, well, what does it mean to be probiotic? What does it mean to ferment? I mean, fermentation is such a deep subject. And so specifically, there are so many different directions I would like to go with you today, but one of them, which we could start with, is this anaerobic digestion of the cow and the rumen, and how they turn this lignin and cellulose and totally undigestible stuff, grass, into the densest muscle and milk and the best food on the planet. So maybe we could start a little bit there about how these incredible biological machines actually function.
Great. If you think about it, you think about what goes on anaerobically. Basically, if you look at the bottom of a lake, for example, or in a sewage digester, where all that carbon has to be turned over and converted back to something, to close the biogeochemical cycle. In these long retention time environments where microbes can just sit there and do their work slowly over a long period of time, you end up with something called mineralization. In other words, all the organic material ends up getting converted to either methane or carbon dioxide. Okay, and these diffuse out into the atmosphere, and they are recycled. There are methane oxidizing bacteria, for example, that utilize the methane and convert it back to CO2. So you've got this whole biogeochemical cycle. But you have basically in these long habitat, or these long retention time habitats, you have basically a complete mineralization, a complete conversion back to the most basic methane and CO2. What's interesting is that this process is very efficient, but it's also very slow. When you look at what goes on in a ruminant animal, you have something that's somewhat different. You have the first couple steps of anaerobic digestion. You have the basic breakdown of all the lignin, and not the lignin, but the cellulose, the hemicellulose, proteins, etc. They're converted down to what are called volatile fatty acids, acetic, propionic, butyric acids. And this is the same thing that happens in a digester, but in a digester, the process is carried to completion. The VFAs are converted to acetic acid and finally to CO2 and methane, but it's a very slow process. In the rumen, we have an incomplete fermentation. So what happens is the polysaccharides, the proteins, are hydrolyzed to their basic building blocks. These are then fermented to the volatile fatty acids, but then the cow steps in there, sucks up those volatile fatty acids, and that's what provides the energy for the animal. If a cow could not subsist on sugars in the form of sugars, they have to be converted to VFA, okay? And this is a process that can occur very rapidly under the right conditions. And so if you look at the rate of, for example, cellulose digestion in the rumen and compare that to an anaerobic digester, it's a 1 or 2 order of magnitude difference. So you could have up to 100-fold faster degradation in the rumen than in an anaerobic digester, okay? So it's a very intensive process, and I think it provides a window on how we might be able to recycle waste more completely and more efficiently and more rapidly, because if we can convert these primary organic materials, cellulose, etc., down to the VFAs and then recover and use those VFAs, that process can run very, very rapidly. And the VFAs contain lots of energy. That's why the cow uses them as a growth substrate, because they contain lots and lots of energy. Probably 80% of all the energy that's in the feed gets retained in the form of VFAs. Okay, so very little goes to waste. Okay, and it's very efficient at fueling the animal. So what we've been interested in over the years is what are the rate limiting steps in this conversion, and what can we do to sort of alleviate these rate limiting steps to relieve the limitation. So we've been focused on that for a couple of decades now.
So when you talk about this process, I had no idea until talking to you and doing a ton of research as to what actually happens when a cow is turning grass into these volatile fatty acids, these VFAs. So, I was kind of fascinated. Every single part is so incredibly specific and energy efficient. Like when they're eating the grass, they don't chew. Why is that?
Well, they chew it eventually. So it's kind of interesting because, if you look at people who want to construct biorefineries, for example, for converting fiber materials, relatively indigestible materials like cellulose, into useful products, they always say, well, you got to do some kind of pre-treatment here because you got to separate the cellulose from the other components. You got to get the hemicellulose away from the lignin. And until you do that, you're really not going to get anywhere. Well, nobody pre-treats the forages for the cows. They find a way to do it. And what do? They use a process called rumination. So as you mentioned, when a cow eats, it doesn't really chew its food. It just bites it off and swallows it. And a typical cow will have about anywhere between about 12 and 20,000 of these so-called ingestive events where they bite something off and swallow it. About 12 to 20,000 per day, okay? But that's not where the action is. The action is what happens after. When it swallows it, what it does then is it carries out a process called rumination. It wets the feed, it rolls it around in its rumen, and then it regurgitates it back up into the mouth. This is the cud, and it chews the cud. And when it chews, it doesn't just chop, it shears. Okay, and if you ever watch a cow, it's doing this. Okay, its mouth is going like this. It's got this circular motion, and what it's doing is it's grinding and delaminating and stripping off layer upon layer of plant material from, say, a leaf. And so what you get as a result is a tremendous exposure in surface area. So we can use an example. For example, we know from experiments that the rate of cellulose digestion is directly proportional to the available surface area. Okay, so if you think of the most familiar example of cellulose is a pad of paper. Okay, a pad of, just a pad of paper. Well, you could increase the surface area by ripping that pad of paper in half and then ripping it again and ripping it again. That's hard. It's a lot of work, okay? Much more efficient, and you expose much more surface if you just take one page off at a time. You just rip a leaf off. You rip the leaf off. You rip the leaf off. And that's what a cow is doing when it's doing that rumination. It's shearing and removing layer after layer and massively increasing the surface area of the substrate. And this allows rapid degradation, because the other interesting thing about ruminal microbes, particularly the bacteria, is in order to digest cellulose effectively, they have to attach to the surface. They're not just floating around there shooting out enzymes. They're actually sitting on the surface of the plant material and releasing the enzymes right at the surface, or even holding the enzymes onto their cell surface, where they do the cutting and the breaking of the cellulosic bonds and releasing the oligosaccharides that they use for the fermentation. Right? So it's a very energy efficient process, because the amount of energy it takes to do rumination is much less than it takes to do, for example, chopping feed in a grinder. Okay, so what you end up with is a low energy process that massively increases the surface area and massively increases the rate of degradation. Okay, that's not really captured in any industrial biorefinery process. And so one thing we've been looking at recently is how can you construct a reactor that will incorporate the features of rumination, do biomimetic rumination within the reactor themselves. Okay, so the idea being that you simultaneously grind, exposing the surface, let that be colonized, and let the fermentation continue, all within the bioreactor. Okay, and this is, I think, a real promising way to improve any type of cellulosic bioconversion, not just with ruminal microorganisms, but anything in a bioreactor that, for example, digests cellulose, ought to be able to benefit from this sort of what Leland at Dartmouth calls co-treatment, where you're fermenting at the same time that you're doing a physical treatment that will expose additional surface area. So I think it's a very promising field of research.
Yeah, it seems like it makes sense. I mean, to recap what's happening for the ruminant, the cow, when it's eating the grass, it's basically not chewing at all. It eats it, and when you look at the teeth, it kind of makes sense. They're ripping and they're just swallowing, ripping, swallowing, and you said 10 to 20,000 a day or something like that.
Yep.
So a whole whack of stuff is getting ripped and chewed and swallowed. Then in their big rumen, which is a huge organ. I mean, when you look at this thing, it's a fermentation vessel. It's pretty big.
Yep.
And there is this way that it kind of takes the material and works it in this kind of circular way, so that by the time what was consumed gets back to the point of sort of regurgitating it back up to chew the cud, it has been pretty well inoculated. It's, if we look at inserting some kind of spore or microbe into there, it seems like there is something happening, whether it's just hydrolysis or an actual inoculation. It seems to me like there's something happening there. So talk to me about that piece, and then we can, keep going.
Okay, so first of all, to put in perspective, I mentioned the figure 12 to 20,000 ingestive chewing events, which are just biting and swallowing, okay, per day per cow. By contrast, there's about 20 to 40,000 ruminative chewing events. In other words, where the feed comes up and it gets chewed and chewed and chewed. Each one of those is an event. But so there's way more of those than there are of the ingestive swallowing steps. Okay. Secondly, the interesting thing about the animal is that it's figured out a way to do the grinding most productively in the teeth. And the way it does this, it will regurgitate a bolus of food. Okay. This food's now been wetted, and it's much more easy to degrade by, or I should say, to rip apart by delamination and shredding. Now that it's wet, okay, that's one thing. But the other thing it does, it's very interesting, is when it first takes the food up into its mouth, the cud, it doesn't just start chewing, it squeezes it out. And so what it does is it squeezes almost all of that liquid back in, and it takes with it all kinds of very small, tiny particles that have already been chewed pretty well. And so the remaining material in the mouth that's now subject to rumination are only the big pieces that still need to be chewed. The small stuff that would just get in the way of the shearing and delamination, it's already been ground. It doesn't need to be ground anymore. That's all swept away, and all we have left are the big pieces that need to be ground further. It's a fascinating evolutionary adaptation.
Now, this is maybe not directly related, but my understanding is that calves, when they drink milk, it bypasses the rumen almost entirely.
That's
And,
correct.
and it goes right down to their digestive system, which makes sense because the milk is already fully digestible. So
Right.
do you think that this is somehow related to that, or are they just different things?
Well, when the calf is first born, she doesn't have a rumen. She's got tissue that's going to become the rumen, but it takes a couple of months for that rumen to develop. And so initially, the animal, as you say, is fed colostrum or milk, and only over time as that organ develops, does it get colonized with a community that can more effectively degrade feeds other than milk. Okay, and so over a period of time, fiber is introduced with the diet. I should say, the diet is modified so it has a higher and higher fiber content as the calf ages, and that facilitates further development of the rumen. The more fiber in the rumen, the more of a physical signal it is to the animal to start moving things around and to begin the process of rumination, chewing the cud, etc. But that only happens over a period of a couple of months. Okay, and one of the interesting things about calf development is that we view it as one possible way to direct the eventual microbial community in the rumen that forms. Okay, because it's kind of a blank slate when the animal starts. It has mostly lactic acid bacteria and some of the kind of bacteria that you and I have in our own so-called monogastric digestive systems. The rumen-specific microbial communities only develop slowly over time. But what's interesting is when they develop in the individual animal, it is a remarkably host-specific and resilient community that cannot be easily disrupted. We did an experiment about, I'd say 10 or 15 years ago, where we used cannulated cows. These are the cows that have portholes in their side, okay, that have been
Oh yeah.
surgically implanted, okay?
Tell me just real quick, just talk about what that is because I had no idea, and it is a wild thing. It looks a little crazy. Yeah, what is that?
Okay, so what you do in cannulation basically is you, under anesthesia, local anesthesia, you ligate the rumen wall to the side of the cow, okay?
Which is like
So
sewing, I suppose.
yeah, you're putting in sutures. Okay, so the veterinary surgeon will put in sutures, and then once those heal, they will go and cut a hole through the skin and through the musculature, so you have direct access to the rumen, and they will slip in a plastic plug, a plastic collar with a plug in it that once healed will allow you to just take the plug out anytime you want, reach in, grab a sample of rumen contents and take it out. Now remember, the rumen contents it's about 15% solids on a weight basis. But if you stick your hand in there, it feels like mostly solids, okay? There's liquid in there, especially if you reach further to the bottom because you have to go through a kind of a mat on top that's mostly feed. You get to the bottom, it's more liquid, but there's still lots and lots of solids in there. But it is a very enriched environment for microbes because it has a very high density of fermentable material. Okay. Well, one of the nice things about, and I should mention one thing, cannulation is something that's frowned on in a lot of circles. And in fact, I'm told that the European Union will prohibit cannulation of cows or animals in 2026. And I think this is an unwise decision. When you want to sample the rumen, you want to do it want to minimally stress the animal. If you do proper cannulation surgery and proper post-operative recovery, you have now an animal that is very comfortable with having its rumen sampled very, very frequently. The alternative to rumen sampling is what's called oral stomach sampling. You basically ram a tube down the animal's throat, and then you have to pull out the rumen contents. You don't get a representative sample because it's hard to get the big solids out, okay? And you get contamination from saliva and everything else. And it's also very stressful for the animal. You know, it's true that it doesn't involve a surgery, but it's uncomfortable for the animal and it's never welcome. I've never seen an animal open its mouth and say, sample me, sample me. Whereas if you have a properly cannulated ruminant, you can walk up anytime, pull the plug, stick your hand in, and the animal will basically not respond. It's no stress to the animal. Okay, so I'm a big favor of cannulation. The disadvantage of cannulation is if you want to do studies with large numbers of animals, well, you can't really be cannulating hundreds and hundreds of animals. Okay, was if you have a stomach tube, you can go in and sample as many animals as you want. And I guess if you want to do population studies in large numbers, across large numbers of animals, okay, I can see where oral sampling is a way to go. All right. I should also mention now that there's been considerable work showing that if you just swab the animal's mouth after they ruminate, you get a pretty good approximation of what that rumen contents is. So I think that might actually be the future in terms of long-range studies where you're studying large number of animals. Okay. But anyway, back to the story. Cannulation, we took some cannulated cows. And if you ever stick your hand in the rumen, the first thing that happens is the cow will sense that there's more in the rumen than there was before, and they'll think it's food, and they'll start squeezing your arm, okay? So they'll put a clamp on your arm, and you wait a few seconds or a half a minute or so, and it'll let go of your arm, and then you can kind of move it around, all right? And you can grab in the rumen contents, pull out samples that are both solids and liquids, and use them for various studies. You can determine what the microbial composition is. You can determine what the concentrations of the different metabolites are, the VFAs, etc. The other thing you can do, and what we did in the experiment, is we asked ourselves the question, how individualized is the microbial community in the rumen? How similar are they from one animal to the other? So what we did, and then the second question is, how plastic is that? Can we move that around, or is it something that's very, very specific to the animal? So what we did is we took pairs of cows that had similar rumen chemistries. In other words, their VFA concentrations were roughly similar. They were a little bit different. But they were getting the same feed. They were standing next to each other in the barn, same environmental conditions. What we did is we had garbage cans, and we emptied about 95% of the contents of the rumen from each animal. And then we switched the garbage cans and stuffed the materials back into the other cow. So basically, you can't get everything out because there's all kinds of little folds and nooks and crannies. But you can exchange most of the rumen contents. And then what we did is, over time, we looked at the rumen chemistry and we looked at the microbial communities. And what we found, to our surprise, is that the rumen community, the microbial species and their relative proportions, went back to the original composition within a matter of just a few days.
Wow. See,
So,
this is the central thing, is that there is some modulating factor inside of the cow that is determining the community.
yes. And what's also interesting is I mentioned there were some differences in the ruminal chemistry, the VFA proportions and those sorts of things. Those proportions returned to their original composition within about a day. Okay? So what it means is the cow had a lot of control over her own rumen chemistry. The rumen microbes may be producing the VFAs, and they may be producing ammonia, etc. They may be setting a particular pH. But then the cow steps in, it decides how much VFA it wants to absorb, how much it wants to pass, and it brings that chemistry back to where it was most comfortable with, within about a day. It took longer for the community to come back. It usually took about 2 weeks, okay? But still, it came back. So here's an interesting question then. If I want to modify that community, how am I going to do it? Because if I can throw in a probiotic, and people have done many experiments over the years where they've dosed particular bacteria or groups of bacteria into cows in large doses, and they come back, and within a few days, those bugs are gone. They're not there anymore. What happened? Why don't they persist? Okay. So one of the feelings right now is that this adult rumen microbial community is fairly stable and not easily perturbed. And if you want to modify that community, you're not going to be able to do it once that community has been fully established. So there's been a lot of work of late where people try to do early intervention. They try to dose calves, for example, who haven't had a fully developed rumen yet. They try to dose them with organisms that they hope will stick over the course of time. And those experiments, by and large, have not been particularly successful. So getting the community we want is not going to be terribly straightforward, even if we knew what that community, what we wanted that community to look like. So this is a big challenge in rumen microbiology: is how do you intervene to establish the microbial community that you want?
Do you have any working hypotheses around what factors are the prime movers when it comes to setting and maintaining these complex microbiological environments?
Well, certainly the cow must be involved. The cow produces lots of growth factors, hormones, and things like that apparently have an influence on what the community ends up looking like. And some of these components, some of the contributors here, seem to have a genetic underpinning. In other words, if you look at the whole genome sequence of the animal, and the whole genome sequence of the bulk microbe of the, I should say the whole genome sequences of the bulk microbial community, the metagenome, the microbial metagenome, and you look for correlations, people have found relations between specific genes and specific microbial community members. And so it's thought that there must be growth factors or other sorts of signals that are being transmitted, crosstalk between the animal and the microbial community that ultimately settles what that community looks like. You can kind of think of this whole notion of what happens when you insert some organism into the rumen. You can kind of use like a sociological analogy. If you think about maybe like say a schoolyard where you got a new kid in school, all right? That kid has to penetrate into that community, has to integrate themselves into that community. You've got people there who have no interest in that person integrating successfully, your bullies, your cliques, whatever. And these organisms will prevent some of these other newly introduced organisms from establishing themselves, particularly if they have to compete with them for something. Okay, so so the number of organisms that might not want the newbie to establish themselves probably greatly exceeds the few other organisms that might benefit from that new organism coming in. So it's a stable community that is not easily perturbed under normal circumstances.
So, this to me seems like one of the key unknowns that we have in biology. I mean, if we figure out how animals manage microbial populations, that's the key to biological manufacturing at scale. So, in terms of the organisms, there are a lot of different kinds. Do you have some intimation as to what kinds of organisms are driving, or is it really like, it sounds like you think it might be genome based, that it's the genes that are driving?
It's genes and also expression of those genes. You know, you not only have to have the right genes, but they have to be able to be accessed and exploited and expressed under the right set of conditions. You know, it's kind of like, you know, it's a classic problem in microbial ecology and really in biology in general. The old Roman poet Horace once said, he said, "You can drive nature out with a pitchfork, but it will always come back." Okay, so, and in fact, I always have this close to my computer because I love the entire quote. It says, "Drive nature off with a pitchfork, she'll still press back, and secretly burst in triumph through your sad disdain." The idea being that no matter what you do, nature's going to push back. Okay, and it's so it's just it sort of encapsulated. It sort of encapsulates the whole issue of how do we alter the rumen microbial community. Okay, so you know there's that, and then the other thing is, you know, I like to think of the microbial community not just as something that's stuck in the cow and is just going to be doing its work there in silence forever, but something we can exploit for our own benefit outside of the cow. And so we talk about what we call extra-ruminal fermentations outside the rumen. And if you think about it, you have a very stable community, okay? And it produces this mixture of volatile fatty acids, which you know are highly energetic compounds. And they're also compounds that, when isolated, are capable of being used as feedstocks for production of other chemicals. Okay, so we can envision a system, and other people have done this, although weren't thinking of the rumen when these initial ideas started evolving in the 1970s, of what is called a carboxylate platform. And the idea here is to use carboxylates, which are basically volatile fatty acids, as feedstocks for industrial fermentations, and you produce these carboxylates not with pure bacterial cultures, but with mixed microbial communities that are good at making VFAs. And there's no mixed microbial community better at making VFAs than those from the rumen. Okay. So you can take rumen contents, we can put them in a reactor, and they will produce VFAs just as happily from a host of organic compounds as they would when they're in the rumen itself. Okay, so they're very durable. It's a very durable community for industrial VFA production. There's two problems, okay? And in fact, I should mention one more thing. People have used the carboxylate platform or researched the carboxylate platform using organisms primarily from other environments, from ocean mud, or from anaerobic digesters, okay? And all of these have the problems is, yeah, they'll make VFAs, but they want to keep going and make methane. So unless you want to throw an inhibitor in there, they will mostly make methane and not VFAs, okay? And they'll only do it slowly, all right? In the rumen, not as much methane is made. Now, you hear about this all the time, cows belt, they erupt all this methane, they're contributing to greenhouse gas. It's true, but actually, they only lose about, usually anywhere between 2 and 12% of their gross energy is lost as methane. The rest is conserved as VFAs, okay? Then the question, well, why don't cows make more methane? Why don't they do complete anaerobic digestion? Well, the main reason is that cows have what we call a rapid retention time. The rumen is a rapid retention time habitat. Material flows through there rather quickly. And it turns out that during conventional anaerobic digestion, what has to happen is the polymers must be degraded to basically to sugars or to amino acids. And then these get fermented to VFAs. And then the VFAs get further converted. And the organisms that do that further conversion, that convert things like butyric acid to acetic acid, or organisms that convert acetic acid to methane and CO2, they grow very slowly because it's a very non-energetic process. You don't get much energy out of it. And so as a result, because they grow so slowly, they're perfectly happy in a digester. But in the rumen, they would get washed right out before they could grow. The rate of passage through the rumen is faster than the doubling time of the bacteria that carry out those later stages. They get shoved right out.
Wow.
Okay? So in terms of an industrial process, we can envision one in the carboxylate platform where you're specifically using ruminal microbes as the agents to make the VFAs. But there's a slight downside to this, and actually not so slight of a downside. And that is, in the carboxylate platform, you want to not just make VFAs, but you want those VFAs to actually link up and become longer. It's a process called chain elongation. So you want acetic acid to go to butyric acid to caproic acid. You want propionic acid to go to valeric acid to heptanoic acid. Okay, C3, C5, C7, C2, C4, C6. Okay, because those higher VFAs are easier to recover, and they're more useful as feedstocks, not only for chemical synthesis, but they're also more useful for conversion to fuels. Okay? The rumen microbes are terrible at chain elongation. They love to make short chain VFAs, but they're not very good at making long chain VFAs. Now we can intervene, we can throw in electron donors, reductants, that will stimulate that process, but they're still slow compared to the original rate of the production of those short chain VFAs in the first place. Okay, so this is one of the challenges in using rumen microbes in the carboxylate platform is we have to make them better at chain elongation.
So, question for you. When we look at, I'm still so confused as to how this cow is modulating this action, because you picture, your school analogy is a good one. Somehow, the rumen overlords have the ability to expel students that they don't like. They have the ability to enroll students from other schools. Somehow they have modulating ability. And, even when, bad apples come in, they shed them. Like you were talking about, an entire rumen, you would think that level of inoculation or changeover would radically change the population permanently. I mean, you're talking about switching schools, but then through some process, after a certain amount of time, and it's not very long, a few days or weeks, the same populations are back. So, I mean, what kind of signals are happening? I need to know more.
I think we all do. I'm not sure how much the cow itself is contributing or whether it's the microbial community, all the complex network interactions among the different community members. Some members produce compounds that other members utilize. Some members produce antimicrobial factors that inhibit some organisms but not others. Okay, you can think of these as antibiotics, for example. So there are compounds called bacteriocins, which are basically small protein molecules that have antibiotic-like activity, and they tend to be very specific in terms of who they act upon. You may even have strains of one particular bacterial species that produce bacteriocins that inhibit other strains of that same species. So there's all kinds of granularity in terms of the level at which these inhibitions can be carried out. All right. The cow probably has some kind of impact, but it's the community itself that probably does the self-policing to get where it wants to be, with some assistance by growth factors or whatever, crosstalk with the animal. Okay, but when the original studies were done, they were almost frankly unbelievable. A fellow named Vince Verrill in the 1970s, working at the U.S. Meat Animal Research Center in Clay Center, Nebraska, he did one of the first massive dose experiments where he had cannulated cows. Okay, and he partially emptied their rumen contents, and he dumped entire fermenterfuls, 20 liters sometimes, of a mix of buffer and a fermentation broth of one particular organism that was very good at digesting cellulose. And traced, using the very primitive tools of the time, traced that particular organism over time, and within 24 hours, it was gone. Gone. Okay? He had another strain of a highly cellular bacteria that actually came from a pig intestine, but nevertheless had tremendous in vitro cellulose degrading capabilities. He dumped the same organism into the other cows. Same thing happened. Disappeared. Okay? So I don't think the animal can act fast enough on that invader without the assistance of the other members of the microbial community that competed for the nutrients that animal needed.
Yeah,
But
I mean,
but
that makes sense.
it's one of the big unanswered questions right now in animal in ruminant science is how can we modulate or control the microbial community? Okay, it's just right now it's kind of a big unknown.
I mean, that seems like the central question to me too. It's so a friend of mine does some primary research with protists,
Yes.
and he is of the opinion that protists may be one of the key modulating organisms within these communities. Do you think it could be as simple as one of these archaea or populations taking the lead?
Well, I think I do agree with your friend that protists are underappreciated. In general, we tend to think, from a mass flux standpoint, that protists are bad because what they mostly do is engulf bacteria. Okay, and so all the hard work that goes into producing bacterial protein, okay, and remember that unless an animal is fed a really rich, high protein diet, half or more of the protein that the animal gets actually comes from microbial growth, where the microbes convert the carbohydrate substrates plus ammonia to protein, and that protein passes through the rumen, passes through the other components of the digestive tract until it gets to the abomasum, which is like our stomach, and then those bacterial cells are acid hydrolyzed, the proteins hydrolyzed to amino acids, and then downstream the amino acids are absorbed. So we think anything that reduces microbial protein yields got to be bad for the animal. But in fact, the protists are there for a reason, and they actually do have some very good functions. One thing they do, for example, is they're very good at gobbling up starch. Okay, Now, modern dairy rations are very high in starch because it's a high energy compound. Okay, it leads to fast growth. But the problem is that if you feed too much starch too fast, what happens is bacteria that ferment starch go crazy. They make huge amounts of lactic acid and drop the rumen pH to a very low level, and the animal becomes sick. It has lactic acidosis. Rumen protozoa are very good at sopping up starch grains, engulfing them, and then fermenting them more slowly, releasing VFAs, and therefore modulating that acid spike that would ordinarily occur if the protozoa were not engaged in that activity. So protozoa certainly do have some good functions. Also becoming more appreciated is the fact that protozoa are also more engaged in degradation of fiber, particularly cellulose, than they were previously given credit for. And so they're also contributing to fiber digestion, which of course is the crux of why ruminants are so valuable to humanity in the first place. I mean, if you think about it, you think about pigs, you think about chickens. Yeah, they're very efficient from the standpoint of pounds of food, pounds of meat yielded per pound of feed, right? But the problem is they're basically eating the same foods we do. You know, you know, you know. The thing about a ruminant is it can degrade these very recalcitrant things we can't. They're grasses, whatever. And also, and people don't appreciate this, most people don't appreciate this, ruminant animals are a tremendous way to balance out the food production system in that they can utilize all kinds of food waste materials that we would have to dispose of by, for example, anaerobic digestion or landfilling or something. In other words, we're talking about wheat nidlings, we're talking about corn gluten feed and corn gluten meal, things that are byproducts of the human food production industry. What do you do with the massive tonnages of these stuff? Well, the easy thing to do is to feed them to a ruminant animal. Some of them are digestible enough. You can feed into pigs, you can feed into chickens, but a lot of them aren't, and those are the ones that go into ruminant diets, and they help balance the kind of the whole food industry's mass flux of materials. Okay, so people need to think about that when they think about how evil cows are, for example, for the environment. No, they're very important, and they're not maybe as evil as some people think.
That's something that I didn't consider, but
Yeah.
I do think about. Like if you're gonna have food, you should be looking at the nature cycle. I mean, with plants, it makes sense. We breathe oxygen, they breathe CO2, and this perfect cycle. Similarly, with ruminants, they are producing incredible food from something we can't at
Yeah.
all interact with. So
Yeah.
yeah, that totally makes sense, and I think that's sort of an uncomfortable truth. And I tried pescatarianism for a year, and I've really looked at my consumption of food. But it's really hard to argue with the ruminants in terms of a real sustainable connection.
Yeah, and some of these problems too are not totally inside. First of all, there was a study done. Hugh Capper Cornell did a study. This is probably a dozen years ago or so. What she did is she looked at what the actual ecological footprint of milk production was. Okay, in other words, how efficient have cows become over the years. And it turns out that now the actual amount of things like greenhouse gas emissions stuff are way lower than they used to be, despite the fact that we have more and more milk production. Okay, and a lot of this has to do with efficiencies just gained with the animal. If you look at the number of dairy cows in the United States compared to, say, 1945, we have fewer than one-third the dairy cows that we did in 1945. Yet we produce like 3 times as much milk. Okay, because each cow has become an absolute machine at making milk. Okay, now part of it is the cows are bigger, and I mean not just bigger, but way bigger. We have some cows in our herd at USDA that weigh in at a ton. Okay, so not a little lady by any means. Okay, and these cows produce tremendous amounts of milk. Okay, so that's part of the reason dairying has become more efficient is just that we have fewer animal units. Okay, and they are also more efficient at converting feed. Okay, and also we give them diets that are more amenable to digestion, more easily digested than in the past. Okay, when ruminants evolved, millions of years ago, they evolved. They were thought to have evolved really originally as forest animals that ate plant material that wasn't a whole lot different than what other animals ate. But over time, as grasslands developed, the number of ruminants increased dramatically, and their size increased dramatically as well. And so you have animals now that basically evolved under conditions where there was large amounts of poorly digestible forage, grass, and these sorts of things. But over a relatively recent period of time, we've modified their diets by feeding them rations that are so different from those under which they have evolved. Okay, and this has led to a lot of animals that are kind of on the edge in terms of health. They aren't as healthy as they could be if they were fed diets that were more akin to what their ancestors grew up with. It's interesting that if you look at, for example, the longevity of dairy cows. Okay, dairy cows will live a long time if we let them. But what happens after just a couple of years on these rather hot diets is we kind of burn them out and they lose production. They're still very productive, but they aren't as productive as they were last year. And so what they end up doing is culling them. Okay, that's a bit of a tragedy. If you look at a cow-calf operation in the beef industry, for example, you can have a cow out on the range. She's churning out a calf every year for 15 years. Okay, there's no 15-year-old dairy cows. All right, a lot of times in many production situations, 2 lactations, 3 lactations, and they're done. They're sent off. They become cutter cows. I think that's a tragedy. Okay, but it's kind of the way the industry runs. Okay, and it's all based purely on economics. Okay, they've got to get the absolute maximum production out of a cow. They feel they do anyway. And so as a result, the cows don't have that longevity they used to. Also, these bigger, heavier cows that have a lot more, that are fed diets that are not exactly as nature intended, we often have trouble breeding them back, getting them pregnant the next time around. And so, fertility issues are also coming into play here. So, there are issues that the industry has to address. But I don't think we're going to get around the fact that we're going to have cattle with us for many years. Okay, they've just been too useful and continue to be useful, particularly as a way of converting relatively indigestible material into products that we can use.
So I think you touched on a lot of really interesting points there. One of them is that there does seem to be this disconnect with our modern society between ethical solutions and solutions that are pecuniary in nature. So things that make us money versus things that feel like the right thing to do. And by optimizing to the extreme, the money side, it seems like these solutions that are ethical are non-economic. They just can't make you as much money because we have optimized for this. And you can see that from a production standpoint, if you're only going to get 3 lactations out of a cow, then that's your cycle now. And it becomes this kind of awful thing, this killing machine where, it doesn't feel quite as homey as the old farm where Betsy was a part of the family and she gave you the milk, and it was this kind of, circular thing where you took care of her and she took care of you. It seems like we're moving towards this system in general on all sides that is more exploitative.
Well, it's true, but there's more to it than that, in the sense that agriculture changed in many ways. And one thing now is that you can't have that ma and pa dairy like you used to. When I was working in USDA, one of my colleagues, Dave Mertens, who's a very highly regarded expert in fiber digestion. Okay, he's one of the world's expert in fiber digestion in cattle. He's retired some years now. He told me a story. He grew up in a dairy farm in Missouri, in the Ozarks of Missouri, in the 1960s. Okay, and he went from there to study animal science at Cornell University. And when he was there, he met a lady, and they got married. And when they got married, his parents drove up from Missouri. They drove from Missouri. They had somebody watch their cows, milk their cows. They drove from Missouri to Ithaca, New York, okay, attended the wedding, didn't even have time to stay for the reception, drove all the way back. It was the first time in 30 years they had missed a milking. People don't want to do that anymore. All right? People complain about factory farms. The bottom line is factory farms are there because nobody wants to do dairying the way it used to be done. Okay? They want a life. They want to go places. They don't want to have to be on the farm twice or three times a day to milk their cows every day, 365 days a year. So you hire people who come in, crews that will rotate in, milk your cows. You manage the farm, you can get away. You hire a farm manager if your farm's big enough. And you get away, you have a life. Okay. That's where agriculture is going now, at least milk, at least in dairying. Okay. And I think it's long-term better for people because you're not going to get burned out. You can actually have a life. Okay. So I think people forget that. And, the question is, they think the conditions on farms are so awful for the animals. And in some times they are, and there's certainly cases where there's animal abuse and animals not being taken care of. I wouldn't want to pretend otherwise. On the other hand, I think you have to understand every farmer, every dairyman, for example, knows their cows are an investment. They're an expensive investment. And I think they take as good a care of them as they can in a way that they can still make a profit on the farm. Okay, so I think dairying's changed, and in some ways it's better, but in some things like cow longevity and these sorts of things, yeah, maybe not so good. Okay, but there are trade-offs.
So, have you been keeping abreast of some of these microbial solutions to things like milk production, where they'll be reacting some kind of bacterial strain that is making, if not biologically identical, then very close to a milk protein product that is usually powdered and then sold on the market. There are a few of these companies out there making these analogs in fermentation vessels. What are your thoughts on this?
Well, I haven't really followed it closely. I think there's a parallel probably with kind of Beyond Meat stuff where they make meat and they're making it taste like meat and look like meat because they have an engineered hemoglobin, that kind of mimics that's in meat. There's no reason you can't do that sort of thing with dairy, except I think scale. Okay, if you look at the massive scale of dairying and how much milk is being produced, I think the idea that you're going to be able to replicate that in a laboratory or in a fermenter is it just the numbers just aren't going to be there, and it's kind of like the Beyond Meat kind of situation where yeah, you can make a product, it tastes good. I've had it. I thought it was great. I was eating. I thought, I can't believe this. This is really really good. But then you think, okay, is there ever going to be a way that they're going to be able to produce enough of this stuff, not just cheap enough, just enough of the stuff, period, to supplant meat production as it's occurring now? And I just don't think so. I don't know how big a plant you'd have to have even to make the slightest dent into the total amount that gets consumed worldwide by 8 billion people. I don't see it. So, but in terms of the quality of the material, yeah, you could probably make stuff just as good. There was a bit of a debate about this when bovine growth hormone came out, because that was a recombinant protein that was produced by an engineered strain of E. coli, and it was chemically indistinguishable from the natural growth hormone, that cattle produced. Okay, and people were all up in arms, and it was engineered, et cetera, et cetera. Well, yeah, it was engineered, but it's chemically indistinguishable. What's the problem? I don't really see the problem. The bigger problem is, do you want to be putting the hormones into the animal in the first place? That's the larger issue. It's not whether or not it's the same material. Okay. I think the trouble a lot of people had is they didn't think we should just be shooting up cows with hormones. Okay.
Which is a fair thing. I mean, we
Well,
look at
that's a defensible argument. Okay. But the
yeah.
idea that there's something wrong, inherently wrong with this particular protein that's chemically identical just because it was produced in a bacterium, I don't see I don't think that's a defensible argument.
No, you know
That's
what? I agree. If you look at cheese,
Mhm.
um, it's only because we have, I mean, not only, we could also do it the natural way, but, before we had fermentative analogs to the rennet that
Mhm.
comes from, I mean, this is a story that maybe I'll get you to tell because I find it so incredibly fascinating that our husbandry of animals and the necessity of essentially culling the male calves is kind of what leads to cheese being a realistic thing because you have these extra uh, stomachs, basically. So, do you want to tell a little bit about how cheese is made and how that animal husbandry led to our discovery of rennet in the first place?
That's actually a little bit outside my area. I think you pretty much encapsulated it. You get rennet basically from calves. Okay, and these are called calves. Okay, and they're male calves because you're not going to call the females, right? Because they're the ones that are making the money. The male calves get culled. The rennet goes into you put it in with the milk. You stir it up. You heat it to just the right temperature. You put in salt and some other things, and you let it sit there a long time. It coagulates. You squeeze it through cheesecloth, and here's your cheese. Okay, um, it's just mankind always finds a way to find a use for something, and I mean there's a little bit of an analog like if you look at the petroleum industry. Okay, when people started first working with oil coming out of the ground, what do they do with it? Well, they use some of it for they distilled some and they used it for lamps. What did they do with the rest of stuff? They just threw it out, and over time these different fractions all found a use. Okay, somebody say, well, okay, we've modified the process so we make less of waste here, but we still got all this. What do we do with this junk? And okay, we can make asphalt. They can do all kinds of stuff, and it's a little bit like that with agriculture as well. You always find a use for something that looks like a waste. Okay, manure going to a digester to make methane. Okay, or we don't call it manure anymore. We call it um, what's the euphemism? I don't know. It escapes me. It's
Biosolids or something? I don't
well,
know.
no, it's what we call it nutrient management. Okay, when you're disposing of manure, right? And you put it back on fields, right? Okay, because yeah, it's a really good fertilizer, and the farmers are very clever and they find ways to use stuff, and I don't see that's ever going to change. But I think what we need to do is recognize the fact that the challenges are bigger now because there are more people consuming more animal products, and the volumes of these so-called waste streams are getting bigger and bigger, and it becomes incumbent upon us to find the most efficient use for them that we can. Okay, so that's
Yeah, no, absolutely. Yeah, on that piece, it is just it's just kind of crazy to me because the necessity of culling the male calves, because otherwise you just have way too many, is directly what leads to this cheese thing, which we just love. And if it wasn't for the culling, you wouldn't get the cheese. It's just this crazy thing where it almost looks premeditated. Like it's too perfect how well it works out. And your thing about oil and gas makes sense too, because man, like the things that we make from oil, like I would love to look into that more because it becomes this invisible thing. You know, it's just a petrochemical, whatever. But I would love to do a deep dive into that at some point as well. But in terms of, where you see it going, back to the microbiology thing, I mean, I can see a version of the future where both centralized and decentralized happen. I mean, why couldn't we have a little composter that's actually either an anaerobic digester or maybe it's something in between? Like maybe as you're talking about the Krebs cycle or the what's the other one you mentioned?
which I'm not sure what you mean.
Oh, the cycles to produce the VFAs.
The carboxylate platform.
Yeah, the
Yes,
carboxylate platform. I mean,
yes.
I could imagine a system where you mow your lawn and then you just dump this into your little carboxylate, you know, rumen
Right?
analog, and
Yeah.
it makes stuff for you.
Well, you'd have to think about, okay, I'm running a little fermentation right here in my backyard in my little food waste slash garden clippings, whatever, lawn clippings. What do I do with that? Well, you'd almost have to get a system where you'd have a truck coming by picking up from, just like the Amazon truck coming by. You have somebody coming up picking up like picking up your garbage, except they pick this up into a tank or take it somewhere where you could process it and recover the VFAs efficiently. You're not going to be able to do that at home, okay, because there'll be a
There's,
substantial technology there. Okay,
there
but
does seem to be a minimum size where these things have viability.
right, and so what you have to have is sort of like a distributed system where you get production at relatively small scale, uh, post processing at a somewhat larger scale, and then finally the ultimate use of it at a larger scale yet. Okay, so in other words, a facility that recovers the VFAs, and then those VFAs could then be used at a different facility in a chemical synthesis platform for making all kinds of useful chemical end products. Okay, but you're not going to do that in your backyard. Not all of the steps. You're not going to be vertically integrated, as it were. Okay, but that doesn't mean that we can't consider, how can't consider ways that this might be made to happen. So,
Yeah, I can see that as well. I mean, we're already doing a lot of the steps. Like we already have trucks coming up to pick up, compost and whatever. I mean,
yep.
instead of it just going to make compost, it does seem like we could come up with higher value add than compost.
Yeah, and you don't know what the future holds. You don't know what's going to happen to the price of availability of fuel in the future. You don't know whether electrification is going to displace gasoline, those sorts of things. You can't really predict. I mean, there's certainly efforts being made, and it varies by country and by region and by even local at the local level. How attuned and willing the local populace is to doing these sorts of things, because they all require more effort. It's a lot easier to flush something down the toilet than it is to take it out back and put it in a fermentation vessel. Okay, so there's the activation energy that's required to get people to actually do something that involves work, even if the environmental social benefits may not be enough, to say the least. If it was that easy, we'd already be doing it. Okay,
Yeah.
so yeah, there's behavioral modification that would be what to have. Remember Dick Cheney, Vice President Dick Cheney? Many people, myself included, weren't real high on his politics, but he did make an observation once, and he said once that conservation is a personal ethic. Which is right. It was a backhanded statement in the sense that I don't want to do it, but some people want to do it. But those people who want to do it are going to do it regardless of whether or not the government's going to step in to help them. Okay, and there's a certain amount of truth to that. Okay, I think there's only so much behavioral modification you can force on people and still have the whole system work. Okay, so people need to, I think, have more of a conservation ethic in order to make certain things happen. So,
Yeah, I agree with that. That makes sense. I mean, when you look at beer bottles and cans that get taken into the LCBO or whatever, um, it works because even if the people who are primary consumers of that alcohol don't care enough to take it back, somebody might.
somebody
You
does,
know,
yeah, yeah,
somebody does.
yeah.
So, okay, this is not related to this part, but going back to our microbial populations for a minute, horizontal gene transfer is
Oh, yeah.
something that I don't know a ton about, but oh my God, it's interesting. This idea that we can, instead of genetically passing down genes, that we can just like hand them over to our, friends. Talk to me a bit about the role of horizontal gene transfer and what your thoughts are and anything about it.
Well, okay. First of all, mechanisms by which it occurs are fairly well established at this point. I think the one thing that's really come to the forefront in the last few years is just how widespread this has been and continues to be. It's a major driver of evolution, and it's a major driver of microbial ecology in the sense that it allows reassortment of genomes in such a way as to to introduce new capabilities into processes and environments that otherwise would have happened much, much more slowly. Okay, so we can certainly exploit it when we're talking in simple systems with pure cultures, and all the early work in molecular biology where they were doing sticking genes on plasmids and then inserting them into a closely related organism, and now they would express the genes on the plasmids, and now they could do something they couldn't do before. And that's really cool. And yeah, there are situations where this has been exploited for industrial purposes. Doing it at the whole community level in a complex microbial ecosystem is going to be it's something that's going to it's happening all the time, but it's something that we will have difficulty stepping in and controlling or making happen. I think because the systems, these complex systems with many different microbial components, it's going to be very hard to orchestrate any kind of directed horizontal gene transfer. But as I said, it's something that seems to be occurring very well on its own. And if we could select for the outputs we want, the organisms would probably find a way to make it happen if we made it worth their while. And this is one of the big things that I think people don't really understand. We have limited control over what a microbe can do. Okay, and they're a little bit like us. They're acting in self-interest. Okay, if they don't want to do something, if it's not beneficial to them, they won't do it. And I'll give you a good example of that. And this is a rumen microbiology example. There's a compound. There's a type of tree called Lucana, and it grows in the tropics. Okay, and in several continents. Okay, and it produces an amino acid, a non-protein amino acid called mimicine. Okay, and it turns out mimicine is toxic. It gets metabolized by ruminants into a compound called 3,4-dihydroxypyridone. It's a compound that makes animals sick. It gives them goiter and causes photosensitization, other problems. This is not a good thing. Okay, well, anytime ruminants consume mimicine, they got sick. But somebody noticed that in Hawaii, there was a herd of feral goats that could eat as much Lucana as they want, and they never got sick. So what these workers did in Australia, is they captured some of these goats, they got the rumen contents, and they introduced them into goats in Australia. And all of a sudden, the goats in Australia now could handle as much Lucana as they wanted. Okay, and it turned out this was due to one particular bacterium. It was isolated by Milt Allison in Ames, Iowa. I'm sorry, I got that confused with another one. It was isolated by McGarity in Australia, and it was called Synergistes jonesii. Okay, and what Synergistes jonesii does, it's got a very specific metabolism. It can only degrade two amino acids, arginine and mimicine, and it degrades mimicine to a non-toxic compound. And therefore, as long as the Synergistes organism was established in the rumen of the animal was Lucana resistant. Okay? But think about this. There's a strong selective pressure here. Nobody else can degrade, no other organism can degrade, at least none of it was known at the time, that can degrade the mimicine. Okay? And there's plenty of mimicine coming in. Okay? So there's positive selection of the organism. I got no competition, and I got plenty of food. Why wouldn't they establish themselves? They establish themselves very well. Now you look at other compounds. For example, look at in the southeastern United States, there is a common forage called fescue. Okay, and fescue has alkaloid compounds in it, and these alkaloid compounds also make cattle sick. Okay, the problem with this, you think, well, okay, no problem. We'll just find an organism that degrades these various alkaloids, introduce them into the organism, into the animal, and everything will be fine. Well, the problem is you can't isolate an organism that will degrade these alkaloids. And the most likely reason is these alkaloids are present in the forage, the fescue, at very low concentrations. Okay, so there's not a lot of incentive for an organism to learn to degrade a compound that's only present in very, very small amounts. It's a little bit like some of the toxic, you read about Love Canal and all these different places where you have these Superfund sites where you have these poisonous chemicals that are present in parts per million concentrations, okay, and they don't seem to get degraded. Well, there's no payoff for the organism, okay? If the food source is that scarce and in that concentration that low, there's no incentive to develop an entire metabolic pathway for degrading this material. Okay, so you get compounds like dioxin and some of these that are long, they're persistent poisons in the environment. They work at the physiological level in negative ways at very low concentrations, and the concentrations are very, very low, and there's not a lot of incentive for microbes to develop capacities to degrade them. All right, so when we talk about what can we do to enhance rumen functionality, one of the things you can point to is we have to follow the basic rules of microbial ecology, right? There has to be a metabolic benefit to the organism, to the microbe, or it's not going to happen. Okay, and if there are other poisonous compounds that are present in forages. Oxalic acid, for example, in halogene and some of these forages that are fed out west or that are grazed out west by sheep, for example. Very toxic, but also present in very, very high concentrations. And again, microbes have developed, have evolved to degrade these compounds. And if you adapt your animals slowly enough, they're fine. But only when it's only when the compounds are toxic but present at low concentrations that you run against the problem of not being able to find microbial adaptation. So
how does that connect if we look back at that rumen transplant? So we do this whole transplant. Is it because they're both stable systems doing essentially the same thing that they just go back to normal? Like, I'm just in the situation where there was an opportunity, this indigestible thing, but there was a lot of it, that got incorporated into the genome, into the microbial population. But in this other situation where you had two distinct cultures with a similar environment, there was no real change. Like, they just went back to normal or their baseline. I mean, I guess that really does talk to your point about, is there sufficient reason to change? It's like, it seems like the sufficient reason to change trumps, dosage. So maybe even a very small amount of that bacterium would establish in the case there was an opportunity.
Yeah, and I don't want to pretend that this is a universal law that can't be altered. And I'll give you another example of that. After we did the experiment where we did the rumen contents exchange, we thought, okay, we're really looking at pairs of cows that were very similar in production parameters, how much milk they made and that sort of thing. So we did some other experiments sometime later. What we did is we identified cows that were high efficient and low efficient in terms of milk production. In other words, how much milk did they make per unit of feed, okay? And we followed groups of cows over a period of 2 years, 2 separate lactations, and found groups that were consistently high efficient and consistently low efficient. We took 3 pairs of cows, okay? One was high efficient and one was low efficient within each pair, okay? And we switched their rumen contents, okay? What we found is for a period of about a week to 10 days, the efficiency of the animal tracked with the microbial community. In other words, the low efficient cows became high efficient for a few days, a week, 10 days, and then they went back to where they were. Okay. This happened in 2 out of the 3 pairs of cows, but it didn't happen in the 3rd pair. The 3rd pair, the low efficient cow, okay, became more efficient, and its community ended up being somewhere in between the community before we changed it and the community afterwards. In other words, there was some reassortment there. Okay, so the community was now a hybrid sort of community that had adapted to that animal and had stabilized the milk production. I should point out that in all these cows, they were late lactation, and during the lactation as lactation proceeds, as you go later into lactation, the cows become less efficient because the animal is now pregnant and they're nursing the fetus, right? And so the fetus is growing, and so less energy is going into milk production, all right? So what we had to do is we had to kind of plot and replot where the curves were going and how they changed as we changed the community. So it wasn't as clear cut as one might expect, but there was no doubt that one of the low efficiency cows suddenly became more efficient after the transformation, and she managed to keep that better efficiency, and but had a hybrid community that was somewhat different from both the original and the introduced community. So what we need to do is, we need to look at this. We need to have a better understanding of what it is that determines how a cow comes back to the position she was in before. Okay. What's underlying that? And that's largely unknown at this point. Okay. But it's certainly a topic for future research.
Oh my God, yeah. Are there people doing research into that specific topic now that you know of?
I think there's lots of people all over the world, really. A lot of this work's going on in the UK, a lot of it's going on in China, and some in the US as well. But fortunately, they're integrating it more with the animal itself. In other words, they're tracking changes in microbial community, and they're using techniques that are more advanced than the ones we were able to access. They're not only, for example, looking at marker genes to determine that allow us to speciate the members of the community. They're looking at the whole genomes of the community, organisms of the community, and they're looking at the expressions of those genes. And they're coupling those to looking at expression of genes within the animal itself at the same time. So it's become much, much more sophisticated in terms of approaches and methodologies than we were ever able to do 10 or 15 years ago. So yeah, there's a lot of work going on in this way, and a lot of it is looking specifically at feed efficiency and trying to improve feed efficiency using microbial approaches. So yeah, there's a lot of work going on.
That absolutely makes sense. I'm trying to think of. Yeah, obviously that's going to be the driver. There's an economic benefit there. If you can tune these microbial communities to be more efficient, that's going to be good. But changing them in general is also almost equally interesting, because it seems like we just have, I wouldn't say no clue, but just through this conversation, there's still a big missing piece of how these changes are made, why, what determines whether these changes stick or just don't. What a fascinating mystery.
Well, and the other thing that's that you have to consider is the fact that we talk about species, bacterial species in the rumen, for example. We're talking about hundreds of thousands of species. It's not just a few; it's lots. Okay, all right, and all of these are interacting with other members of the community. So the number of potential interactions is huge. Okay, the other thing is that a lot of organisms that are very, very similar to our eyes, even when we look at their genomes, are actually probably doing stuff very different in the rumen because, for example, if you look at the microbial communities, if you look at individual species that are at elevated relative levels in high efficient cows versus low efficient cows, one thing we find, for example, is that lots of these organisms fall into a group of bacteria called Prevotella. That's the genus Prevotella. Well, it turns out that the genus Prevotella is probably, in terms of numbers, the most abundant genus in the rumen. Okay, there are many, many species of Prevotella and many strains of these species. And what's frustrating to us is you can see that there are certain Prevotellas that are more abundant in high efficiency cows, and certain Prevotellas that are more abundant in low efficiency cows. But when you look at those Prevotellas, what's the difference? They don't seem to be that different from one another. And another thing that's frustrating is not only are there, there are some differences in the genome, but one of the problems with Prevotella is it's what we call a generalist bacteria. It can do so many different things metabolically. Okay, it's not like a methanogenic archaea that does one thing, make methane. Okay, it's not like the synergistes that does one thing, it degrades mimicine. Prevotella does all kinds of things. It ferments a whole bunch of different kinds of sugars. It ferments hemicelluloses. It ferments proteins and amino acids. It does all kinds of stuff. Okay, how do we determine why Prevotella A, that's more abundant in high efficient cows, why is that Prevotella more efficient? What is it doing that makes it more efficient? Is it something that Prevotella itself is doing? Is it something that's doing an interaction with some other organism? We don't have a clue. But the number of potential interactions is in the millions and millions and millions. And so it's going to be a long time before we can sort all this stuff out.
What do you think we need in terms of a technological breakthrough to better analyze these problems?
I don't know if we need a technological breakthrough so much as we need to kind of focus. We need to look backward a little bit. And what I mean is this, if you look at rumen microbiology when I was growing up, when I was in grad school, what people did is they would isolate a bacterium from the rumen and they would characterize it, okay, and they would know something about what it does. Even though they didn't have the genome or whatever, they at least had a pretty good idea of what it was doing physiologically based on what we could get it to do in culture, okay? Now the trend is much more, let's sequence genome after genome after genome, and now we got all this genomic information, but it's been divorced from the individual organism to the point that we don't really know much about what that organism is actually doing. We know what it's capable of in terms of, we know what its complete genome is, and we know what most of the genes do, but we don't know about the organism itself. We reduced it to the point where the microbe is a genome and we don't look beyond the genome. We have to go back and look at the microbe. We know that only about 10% of the bacteria in the rumen are culturable by our current techniques, okay? But a lot of the reason that number is so low is because people don't bother to culture anymore. They just sequence genes. What we need to do now is we need to pull out isolates that are, for example, specific. We need to get that Prevotella, that one Prevotella, or those couple of Prevotellas that are more abundant in high efficiency cows. We need to get them out and study them and learn something about them that will give us some kind of clue as to what they might be doing that we couldn't get from just looking at the genome alone. Okay, so I think that's one thing. We need to kind of get back to some of the early wisdom of the early Rumen microbiologists who managed to find tremendous insights from just looking at the culturable characteristics, the characteristics of culturable bacteria. So there's a lady in Israel, Tamar Zahavi, and she went through an enormously intensive exercise and managed to isolate 23% of the members of a microbial community. Okay, that was a very high number, and also did the genomics on it. Okay, but she'd been able to get some real insights into terms of what a lot of these bacteria are, because some of these bacteria are very rare and difficult to culture, but she was able to do that. And now that we have some of these organisms in hand available for study, I think we can learn a lot more about some of these rare species that may be more important than we think about. We have this tendency to think if an organism is abundant, it's abundant for a reason. There's a lot of them. There are a lot of them because they're very successful, okay, and therefore they must be very, very important. But there's also some organisms that are probably not all that abundant, but are also pretty important. They may be interacting in some way that cannot be reproduced in any other type of combination. And if we could understand that linkage a little better, we might be able to gain some insights there.
That makes sense to me. I mean, if you look at any team in sports or, individuals in society,
Yeah.
You may have a lot of employees who do a certain thing, but
Mm-hmm.
they're only there because of other people. So the numbers don't necessarily correlate to the importance. So
That's right.
That's a really good piece.
Yeah.
Wow.
Something to think about anyway.
So what's next? I mean, if you had a magic ball and you were to say, okay, here's the research I want to see, and I want the results on my desk yesterday. Tell me about a couple experiments that you want people to be doing, or that you want to do.
Well, I'm retired now, so I don't have my lab. So it's more like a wish list. Okay. But I would think what I would like to see are, first of all, as I mentioned, I would like to see isolates of particular organisms that reproducibly are I should say, overrepresented in animals that have particular physiological characteristics. And this could be something production oriented, like it could be something like milk production efficiency, or it could be something weird like ability to survive or ability to attenuate acidosis. You know, some cows, you feed them high starch diets and they go acidotic in a moment. Other cows, much more stable. Okay, what's it about that? What's going on in those cows where it's much more stable? Is it just that they're making more buffer? Okay, or is it something about the microbial community that is more resilient and more resistant to changing, more resilient in the face of, of changes in rumen pH? You know, I mentioned buffer. You know, cows produce over 100 liters of saliva a day. Okay, think about that. Some cows, 200 liters or more. Okay, this is what's buffering. Yeah, imagine that. What's buffering the rumen pH. We don't really know at the community level how pH dynamics affects the microbial community. You know,
Probably huge. Probably
I
huge,
don't
huge,
want.
huge.
Yeah, I think I gave you the impression from these rumen content switch experiments that the communities themselves are pretty stable and they don't change much. It turns out that they change a lot during the course of the day. Okay, and so when we do our experiments, we're always careful to sample at the same time every day after feeding because the communities do change during the course of a day. Okay, what I would like to know is what drives those changes. Obviously, it's different feed availability and that sort of thing, but there's also interactions among the microbes that are at play here too. And I'd like to know what some of those interactions might be. Okay, so you know that's so I guess, and the other thing I would think of is people are always. I shouldn't say people. Animal scientists tend to focus on the entire herd. They want basic guiding principles that are applicable to as many cows as possible. They don't want to hear about or they don't want to study weird cows. Okay. I think we need to look at the weird cows and what makes them weird. And we had a cow, this is many years ago, this is back in the mid 1990s. We had an experiment where we had 4 cows and we had them on 4 different diets in what's called a Latin square, where each cow is rotated through a different diet. So they all get the same diet, just at different times, different periods. Okay. And what we were doing is we're doing the very beginnings for us of looking at the microbial community, particularly the abundance of particular cellulose degrading bacteria. But we were also looking at various rumen chemistry parameters, the pH and the VFA concentrations. We had one cow, and this cow, when we put her on a diet that was high in forage, which should give you a relatively high rumen pH, okay, her rumen pH was something like 5.5, which is really low for a cow. And this was on a high forage diet. Okay. And we thought, my God, when we move her to the really hot diet, she's gonna kill herself, right? I can't imagine how low the pH is gonna go. Well, her pH ended up being in the 4s, okay, which is typically would denote a really sick cow, but she was very productive. She was producing more milk than any cow in the group, okay? She handled it very, very well. Why did she handle it so well? What was unique about that animal that allowed her to produce enormous amounts of milk with at a rumen pH of 4.8? And not only that, her VFA concentrations, okay, in the rumen were about twice those of most of the other cows. So she was not absorbing the VFAs very well. They were building up from microbial fermentation. As a result, the pH was dropping because there was so much acid in her gut, but it didn't seem to bother her. Okay, what is it about that cow, and what is it about that microbial community that would even be producing or even be degrading fiber and substrate that rapidly at that low of a pH? It didn't make sense. So here was a weird cow that nobody wanted to hear about. And when we wrote up the paper and we submitted it to the Journal of Dairy Science, the reviewers went crazy. They said, we don't want to hear about this. You know, they basically said, you know, that they weren't interested in this. So we had to put this concluding paragraph that we have to start treating cows as individuals. We have to start thinking of them individual. What makes individual cows unique? And we posited at the time that the microbial community had a lot to do with it. But it was a hard sell to the community because they want kind of universal answers. But, it's like in the medical literature, case histories are a very big deal. You see them published all the time. You never see case histories published in animal science, at least not anymore. Well, they should be. I'd like to see case studies about weird animals and what makes them weird. Okay? So that's something else that I would think we ought to be thinking about anyway. So, just an opinion.
I love that. I think that that's probably where you're going to find innovation, because you're going to see, well, what's different
Yeah,
about this weird one.
yeah. And maybe it's got an unusual organism in there that can tell us a lot, be really beneficial, have some unusual properties, and maybe even elucidate some sort of new microbial ecological principle. We don't know. Won't know until we look.
Yeah, totally. Man, what an awesome conversation. I am so glad that you were able to make the time, and I would like to do a follow-up at some point as well.
Sure. I thank you very much for having me. I'd love to, love to hear your thoughts and anybody else's thoughts about it. It's a fascinating topic and a fun topic. It's just fun being around these crazy, amazing animals and
Yeah.
their microbial symbionts. So, yeah, all good.
Well, Paul, I look forward to touching base for real, and maybe there's even some studies that we could go and do a little podcast on site. Something live that's happening somewhere. But again, thank you so much for your time, and I really learned a lot chatting with you, and I look forward to the next one.
Well, thank you for your questions. I think they're very thought provoking, and I wish you all the best. Take care, and thank you very much.
Thanks, Paul. Bye.
Goodbye.